essentially whatever community was there. Up at the high end, I would imagine there's an initial washout, right? Because there's growth rate, but that water's coming through like crazy and diluting that growth. Mid-bay, there's nutrient delivery, less washout. There was a peak, and that pulse sort of went by. I know at some stations, the signature seemed to dampen out after a couple of minutes. So it was sort of a point event, and I don't associate it with any toxicity that I know. That won't be.
Other questions?
I've got a few for Roxanne. I'm an oyster farmer and doing it about 20 years. And what I consider winter mortality sounds like your diagnosis of SSO. So I wouldn't be surprised if I have slight tinges of it myself. Does a rain event, like the rain event this gentleman was just talking about, if the salinity goes down for a day, for a tide, would that help destroy the SSO? Because the oysters can handle it, but will the organism be able to handle it? Is that possibly a way to save the oysters?
Well, we don't know if that's true for SSO, but we do know for MSX that if we reduce the salinity, it will kill the organisms in the oysters. And because SSO likes higher salinities, I think it's real possible that if you reduce the salinity, you might be able to kill off some of the Plasmodia in the oysters. It's a possibility. It would be pure conjecture, but I think it's a possibility, yeah.
This has been a really bad winter for ice and for super low tides in all the coastal ponds. I don't think I've ever seen the tides so low so many days, basically in a row. Would exposure to the freeze, it doesn't hurt the oysters, would that also help with the SSO or any of the other diseases you're familiar with?
Part of the problem with the SSO, again, is that we don't know what the intermediate host is. If we only knew that, then we would have a lot more ability to control what it may or may not do. True with MSX also. You know, it could be a fish, it could be a snail, it could be anything, and that's part of the problem. There's just no. So if we knew what it was, we'd go, oh, you know, this year that ice is going to knock out, you're not going to get those fish in there this year, or you're not going to get an overflow of snails that carry it. Then I could say, yeah, but unfortunately I can't.
I have one more question or comment, basically. July, August, September, the heat kills SSO. I'm familiar with QPX, which is a quahog disease that also is pretty much wiped out in July, August in the heat exposure. And we have a Vibrio up and coming problem. And the worst thing in the world is to have our shellfish heat up in the summer when we want to sell them. So there's it's a problem for a shellfish farmer because it looks to me like the heat kind of destroys the organism, but we don't want the heat because it's going to encourage Vibrio, which is a whole other problem. Is that correct?
Well, you're right, the heat does increase the amount of Vibrio in the water, and you're right that does appear to decrease or at least increase the clam's immunity or inflammatory system so it can fight off QPX. For SSO, it's really about sporulation, and that is associated with that May-June time period or that temperature. And it may be a temperature trigger, I'm not sure, but in the spring, when the oyster is kind of revving up its machinery because it's been its metabolism has been very low over the winter, it might have as much to do with the revving up of metabolism as it does with the temperature. I wish we knew more about the disease. So I can't say for sure that it's temperature that's causing that sporulation and the proliferation of Plasmodium, but I'm sure it's part of the picture. So what does that mean? I don't know that the temperature is going to be the most important part of it. It's probably also has to do with the animals coming awake and revving up the rest of its metabolism at the same time. Is why the SSO then proliferates, undergoes sporulation. So increased temperature, I don't think will directly cause a change in the amount of SSO that undergoes sporulation at a certain time period as well, but it might increase the amount of overall disease that might happen, depending a lot on what that intermediate host is, and that's part of the problem. If there's something coming up as an intermediate host that wasn't really here in large numbers before, we might see an increase in this disease also, as we start to see animals redistribute themselves in different locations along the coast. We know it's happening. I don't know if that answered your question or not. Sorry.
Let's try.
Roxana, I guess I had heard maybe your discussion with Marta before, but I wondered if you guys had looked at it like, say, the Narrow River population or the Green Hill population where, they're not fished, and, I just wondered how, could you possibly determine, or would that aid in determining what the intermediate host might be, because, you're not having witches coming and going as far as, our relay work or growers, introducing animals. Would that aid in helping figure that out?
It might, if the SSO disease is there, and we can monitor it and look at, but it's really hard to look for, because you're looking at almost everything. And that's part of the problem, trying to find an intermediate host. You've got this giant, enormous number of organisms to test, and a lot of it is just, feels like you're just spinning your wheels for no good reason.
Or some of the places like Quanticook has a background population and hasn't had much restoration work or any aquaculture yet.
Yeah, if they have SSO there, we could definitely look at some of those organisms that are around them in high levels and see if there's anything in them that might indicate that they were intermediate hosts. And that's kind of the way people are trying to approach it, because there's really no other way. But it is expensive and it's labor intensive, stories. They're real. But it's a good thought. It's a good thought to try to look at something like that that's confined.
Other thoughts? Yeah.
Oh, Dave, you had your slide up there where, I think it was for the Alexandria, I'm not sure which species, but how it had been, it was just found in 3 places where it was originally, and then you found it in Rhode Island, and Thomas found it in North Carolina. Now, Thomas had known about it because of your work up here. Do you think maybe it's in other places, but people just haven't known to look for it, or haven't, like you said, looked at large enough samples to look for it?
Yeah, those are the 3 records from North America. So Balek in the probably late 80s, but he didn't publish it until 95, observed it and identified it by fecal plates on the outside of Long Island, somewhere down along Jamaica Bay in that area. And so that record was in there in his monograph, and nobody else really saw it. And coincidentally, right around the same time we picked up this new Alexandria here, some people in Karim Thomas's lab down in North Carolina picked it up. Most likely it's not just 3 spots. It's a little tricky to identify, so it may be a case that, at least for Rhode Island, maybe it's a northward range extension with temperature change, for example, from Long Island up. But we're seeing, what we see is a combination of where it is and who's looking. And so those 3 places now, we looked pretty carefully and found it. Whether it's in between, nobody really knows.
As far as you know, people, they're not actively looking for it.
No. And this was an unfunded, sort of serendipitous thing. I was doing the weekly examination of Woodford for a couple of reasons, not really to look for Alexandria. It just sort of hit me over the head when thousands per liter show up. It's surprising.
Jim?
Well, I was just thinking about the red tide in New England, and typically we associate red tide and PSP outbreaks, Cape Cod, New Hampshire, Maine. Generally, the waters are cooler than Narragansett Bay or Buzzards Bay, and I'm wondering, since the projections are with continued climate change and continued warming waters, do you think red tide will be receding more northerly into cooler waters?
No.
Okay.
Because, so for example, the Gulf of Maine species, the dominant one, Alexandrium fundans, it sort of peters out at 15 degrees C. So unless the winters get so warm that they never drop below 15 degrees C, you you're still gonna have it. The window may move, so instead of expecting it in Massachusetts Bay in April or so, maybe it'll move earlier. But as long as you get below 15 degrees C and then come up towards 15 degrees Celsius in the spring, you'll get it. For Peruvianum, which is the new one we found in Rhode Island, it generally, the data is limited because a lot of people haven't found it around the world. It generally likes it slightly warmer than Funyuns and tamarins, the Gulf of Maine species. It also likes much lower salinity. It's almost a lagoonal species versus Funyuns, which is an oceanic front species, right? It moves down the coast. The map I tried to show, it starts out along the coast and comes down towards Massachusetts. So with regard to Alexandrium, in southern New England, so say south of the Cape, there's two models, two modes. The regional scale where cells get jetted down from the Gulf of Maine. But now there's at least 1, 2, 3, 4, 5, 6 resident local populations that have been published. Okay, so you're in Northport, Long Island. There's recurrent populations there, whether there's a red tide in the Gulf of Maine or not. Same thing with the Nauset Marsh system down at the tip of Cape Cod. Closures every year, it's a localized population. Buzzards Bay, Perch Pond and Woods Hole, and now we have evidence of a resident population in that little cove in Whitford. So there's two models. The big one that gets all the press, and deservedly so, because it covers hundreds of kilometers, is the big Gulf of Maine red tide. Increasingly, there's these little pockets. What's happening there, I really don't know. It just seems like it's showing up south of the Cape in a different pattern than that coastwide swath.
David, are there plans to look for the cysts and the sediments in Whitford Harbor, since it hasn't been done since '79?
I wish, yeah, I mean, that would be the straightforward and logical thing to look for next, so that would be something to fund. I don't have any funds for that secured at this time, but it would be a great thing to check out.
Go ahead.
No, the thing I was going to add to what Dave said, you notice that where all of these harmful species are found, they're exactly in those premium sites where a lot of aquaculture is being developed. And while we have the longest quantitative phytoplankton time series in the world, Narragansett, it was decided by a meeting of all people of time series some years ago in Croatia. The fact of the matter is that the flora in the open reaches of Narragansett Bay is not a particularly good habitat, unlike all these little cul-de-sacs you're finding where these are prime sites for aquaculture. And the concern here is then that, you know, what's going on, and there's very limited understanding of these particular harmful and red tide species in these areas to be able then to lead guidance as to whether it's an appropriate or potentially dangerous site for aquaculture. There's an absolute need to know about it, and it turns out that Rhode Island is probably the only maritime state in the U.S. That has not spun up a harmful algal bloom program with regard to aquaculture and finfish culture, unlike you can go along the west coast, the Gulf of Mexico, and all the way down here. So I think Dave's numbers here are very, very provocative and sobering.
Actually, this is Barbara Olsen. When the synchronous correlation of SSO occurs, I suspect they are released in the water column. So can we see them and detect them in the water sample?
I would expect we could. That work has not been done. It would take some routine monitoring because it isn't an exact date, so you would have to sample several times for several weeks. But yes, that's a possibility. That would definitely be a possibility.
My question is kind of like a general question. I'm not a scientist, so forgive me if it sounds a little off, but I've been involved with the Narragansett Harbor Commission for about a dozen years. So we went from seeing 1 or 2 leaseholders in Point Judith Pond to probably somewhere around 9 or 10 now, with another, at least another 3 applicants in the process. And so, what are these diseases, do they pose a threat to like the rest of the pond? You know, whether it be shellfish, you know, crabs, quahogs, finfish, because I get a lot of questions from people that know I'm involved in it. Not only the user conflicts where, you know, you're giving up space in the pond to one user, but what is it, you know, what are the threats to the ecosystem of the pond, adding so many animals that aren't there normally, they weren't there 12 years ago?
That's a tricky one. In general, right, think of all these shellfish as filters, right? So they're exchanging particulate phytoplankton in the water column to protein, essentially, that people can eat, which is a good thing. As long as, and this is a little bit outside my real area, I'm in the phytoplankton and things, but as long as you don't modify the pond so much that there's some sort of nutrient buildup effect, say under an intense aquaculture site, there could be a buildup, say, of pseudofeces and a change in nutrient cycle. I don't know what level of loading you need to get that. That is one potential scenario. But I think within, you know, a fairly well-flushed system, as long as you don't hit some level, what that level is, I don't know, is generally going to be more of a beneficial thing for the things most people would assume is water quality, right? The water should get clearer for the most part. So that's more of a shellfish thing, which I'm getting outside my area.
I
Yeah.
can add that, you know, we know from 200 or 300 or 400 years ago, there were, I don't know, 1,000 times, 2,000 times more bivalves in all of our local ponds than there are now. What we're putting out there is aquaculture as a pittance compared to what used to be along our coast. They are filter feeders, and they evolved there for a reason. They've been a very, very important part of the ecology for thousands of years, and I think if anything now we have almost nothing left. So what's being put out there as part of aquaculture doesn't even begin to touch what used to be there. And I think we need them. We are starting to find some interesting information that when we start putting more bivalves out into these areas, they're cleaning up the water. They're helping to take things out that have been problematic for us because we have until now lost our filter feeders. And they are really our little vacuum cleaners. So we want them out there, and want them in high numbers. How will that affect the ecology of those locations? Yeah, probably will. Because you're filtering out some things, and you will have pseudofeces that are produced. But I know that centuries ago, those pseudofeces were there in a whole lot larger amount than they are now, and yet we had a very functioning, wonderful ecosystem. So I think we're way out of balance, and I would not restrict any bivalve culture because it might affect the balance that currently is in place, because I think the balance is currently in place by what you're saying, even at the HABs, is still way out of balance. So what is normal? It's not what we have right now, really. So I don't think it can hurt at all, and it can always, I think, help to have something that will filter out. What's happening with the HABs? I've done a little HAB work on bivalves also, and it's a problem. There are HABs that, like the Eriococcus, will close the animal down, it won't eat, and it can actually die because it can't reproduce, or the spawn, we did some base scallop work, and the larvae cannot eat the HABs, and when they're proliferating in the water, the larvae will die. So there's lots of problems going on with these interactions with all kinds of things out there. We don't understand a lot. There's a lot we need to do.
Yeah.
But I would never limit aquaculture, bivalve. Fish aquaculture might be something else. I hate to say this, but fish aquaculture is something you need to worry about with pollution. Bivalve is like the opposite side of the coin. It cleans up, it doesn't end. So.
But just, is there any threat from these diseases? Is there a threat to like other species, or is it just, do they just go after the oyster?
As far as we know, SSO, MSX, and dermal that we didn't talk about today, those are all diseases of oysters. And in fact, we know that MSX and Transderma were introduced by bringing other non-indigenous oysters to the East Coast. And they didn't cause disease in the other non-indigenous oysters, but once that little agent got into eastern oyster, it went crazy. So, you know, what we really, again, we're looking at spreading animals around and the agents that cause disease inside them when we spread the animals around. Those are more questions and things to look at, you know, where did the HAPs come from? How did they get here, you know? Those so many theories and thoughts on that.
And there's examples not from Narragansett Bay, but for example, the Gulf of Mexico red tide foam used to be called Carenia brevis. Now it's called Carenia brevis. For years it was a problem, say in Florida. People didn't worry about it. But in the 1980s, just by chance, whatever happened, a large population of these cells got caught in the loop current, came around Florida, and suddenly they're a problem in North Carolina. That's become a perennial problem. So it's the interaction of the population dynamics of these phytoplankton, right? They're just doing their thing. They get moved around. With regard to Alexandria, what's different now, it seems, south of the Cape, is that it's not just this regional thing where you worry about, you essentially sit in Rhode Island and say, boy, I hope Massachusetts and Maine don't have a big ear and send it down here. There's localized populations. How big they are, we really don't know. We're just sort of seeing it for the first time. Whether it becomes a big problem, again, another thing to check out. It should be some part of this whole management plan in a way, to know very basic stuff. Try to find a paper on what kind of phytoplankton are in any of the salt ponds down the coast. Maybe somebody's keeping track on their own. Maybe some of the growers are. You would know more than I. But it's not well characterized. It's not well described. It's very, very basic information. That would be great to have. If anything, from this little talk, just that there's a perception that people have looked at Narragansett Bay forever. We know everything about it. Two things. It's a mixed habitat, right? What's happening in the mid-stem of the Western Passage doesn't mean it's happening exactly the same in every salt pond. And the other thing is that the patterns that were described in the 60s and 70s with, for example, diatom dominance of the winter-spring bloom. That's not always the case anymore. A non-toxic dinoflagellate, Heterocapsule triquetrum, is now the dominant, even through the ice in Whitford this winter, cold winter. Numerically, there's more of this flagellated dinoflagellate in that little harbor, that little cove, than diatoms, what you would expect. It's not toxic, it's not a problem, it's actually good food, right? You raise shellfish on it sometimes. But it's just different than what we saw even 20 years ago. So it's a very basic thing to know what kinds of phytoplankton are around and at what numbers. And it's just something we don't have a lot of information on, despite the perception.
I think one of the things that I've talked about with other aquacultures before is to try to be able to monitor what kind of plankton are occurring at different times during the year. And we just haven't had that ability to do that, but I think that would be really good information to have at some point.
And ideally, I mean, we've tried several times, Ted Smader and myself, to get funded for that sort of thing. With some partnerships with growers, they were loose associations, but we did not get funded in these last several tries of that. So anybody who's on the growing end who's interested in that kind of thing, let us know. I think that kind of partnership, if it's helpful information for managers or growers, I know we'd love to help out with that.
Good. Jim first, and then Jim back.
So one last question for you. So we all know the popular shellfish that we like to consume, whether it be oysters, quahog, soft-shell clams, mussels, they're all filter feeders. And whether it's a toxin-inducing dinoflagellate or some other HAB, or bacteria or virus because of fecal contamination, is depuration going to be a management tool for the future? Is that something that you see is going to be inevitable?
I think it's going to be one of the tools in the toolbox, depending on the season of the year and how much flushing there is in a certain area, what the temperature goes up to. It'll be one of the potential tools. There's still some work to be done about how to do that and where it could be done, because these are expensive things we're talking about. But it's possible that that may be one option for certain things.
And I ask that with trepidation, because when I was, shellfishing full-time for a decade, the Shellfishermen's Association in Rhode Island was adamantly opposed to depuration because you essentially don't rely on a free, open, and common fishery of hundreds of shell fishermen working the waters in the state, but now you have a select few individuals that can go into the coves that are presently closed to shell fishing just because of elevated fecal coliform. And it doesn't take that many shell fishermen to produce harvesting the volume of shellfish that hundreds of shell fishermen would be bringing in on a normal basis. So I'm not advocating depuration. I'm just curious as to whether or not you see that in the future as a tool that from a management perspective may be required.
Especially when we start looking at Vibrio and things like that, we might, at certain times of the year, need to think about how we can get, if there's a Vibrio increase, how to get those down and still have a good sellable, healthy product. And I agree, it funnels everything into one thing, but hoping with cooperatives forming and the shellfishermen working together that we can work. I don't want to see one giant company involved. I don't want that to happen either. That's not what should happen with shellfish.
That was called Warren Finn in the 1980s, by the way.
And for the wild fishery, if you had good knowledge of what species were there, for example, the toxic ones, you know when they disappear, right? You're monitoring. You know a depuration time based on temperature in the species. You say, okay, this species that's toxic was around for 3 weeks. Just sit on things for a couple of weeks after that. They should be naturally depurated. Then you can reopen. So it's manageable if you have the basic information about when these toxic things are around, which ones they are, what their processing rates in the mollusks are. And you could, if you have more information, have that kind of strategy. Right now, I'm not sure where that is.
Thank you.
I don't think that the Rhode Island Department of Health has any regulations for depuration plants right now. Personal knowledge, I don't think you're going to be able to, you're not going to be able to slide that through for a long time.
No, they
So I
don't.
wouldn't even worry about that. You were talking about algae blooms. There's a check on the shell. They change. You can oysters and clams. I grow them both. It's real obvious. I can't tell you how many times I've talked to people. This oyster is 2 years old. I'm a scientist. It's 5 years old. Well, I bought the seed. I know how old it is. It's real obvious sometimes when the algae, when it changes, especially with cohorts.
Yeah.
They'll be sitting there, they'll be sitting there, they'll be sitting there, and then all of a sudden, what happened? They're double the size, 50% larger. So it's pretty obvious when that happens. I'd be willing to help you with that. Another comment is, you said about the algae in the water in the wintertime.
Yeah.
We harvested 3-inch oyster. We're out of them in January. We go back a month later through the ice, and it's amazing how many market-sized oysters we find.
It's been.
And we're covered in ice, and we still find growth. It's amazing.
This particular, not this winter, but it was either 1 or 2 winters ago, the abundance of this dinoflagellate, a non-toxic one, Heterocapsa triquetra, off that little dock in Whitford Cove. I looked around, I couldn't believe it. I did the counts 3 times. They were so high. The only place I've seen anything that high was in the Baltic Sea. Millions and millions per liter. I mean, the water was golden brown. I mean, and supposedly that's pretty good food for shellfish. So why that's happening, why they're around so much more now than what we'd expect, I'm not really sure.
We're almost at the 6 o'clock hour. I'm going to take one more question, and I think we can go informal after there. So you've got the
One
last
question.
one. More comment.
This isn't intended to dismiss what Dr. Smeda said, but the DEM Water Resources section has a program where they have I'm not sure who the individual is, who are not directly involved, but they have contracted someone that identifies the algae and looking for the harmful species. And when they have a sufficient concentration, they they tell us the location of their water sample, and then I go get a shellfish sample, and then they look for the ASP or the toxins in those. But I'm not judging its adequacy, because it sounds, your description would be a much better approach, I would think, but again, it's not that it's not being done.
Yeah, it would be great if even zeros, like we went and we didn't see anything, it would be great to have that available. Because right now, and I mean, I've talked a little bit with some DM people, so I know a little bit about it, but I don't know the real deal tables at all. And if you're relying on essentially phytoplankton to support the shellfish that are growing for business, which I fully support, it would be great to have that basic information, whether it's coming from an already existing program or a program that needs to be spun up, whatever. It's important information to get out for all of you. The anecdotal evidence about you saying the color of a big bloom and blooms and bursts and growth, I mean, if you start to do some very basic phytoplankton identification, you'd say, well, here's what's happening. The bloom was early, or it was this species, and you had great growth versus other years, not so much. I mean, it's real basic stuff that if the state's doing it, it's great, but let's make it visible, and if it needs to be beefed up. My impression is that funding agencies are really look favorably upon partnerships, right? So if it's something that has a greater demand, you know, that maybe that's what it needs to push it through to improve it either at the state level or however it happens. But I think it's important. But yeah, I know there's some level of state monitoring, and that's great. But it would be good to have some more info on that.
Well, I'm going to ask you all to thank from Sea Grant for attending this one. We have another one coming up, date not decided other than April. Sometime probably early part of April, going to be a focus on nitrogen and nutrient dynamics and cycling here in Narragansett Bay. I don't know if you two have to run off. I don't know if they have another function in here they're going to move us out for. But if you two are willing, if you want to grab some cheese, cookies, crackers, mingle, and chat a bit until they boost us out of here, that's also fine. Thanks again.